Hi guys, today we're doing a lab with Lactaid and milk. In this lab, I have these different solutions that have been labeled, 4 test tubes in a test tube rack, some test tube holders, and a Benedict solution, which is an indicator for glucose. Also, I have a hot plate with some boiling water, which requires caution. Test tubes have been labeled A, B, C, and D. Test tube A is going to get 2 milliliters of milk and 1 milliliter of the enzyme solution. Test tube B is getting 1 milliliter of milk, 1 milliliter of vinegar, and 1 milliliter of enzyme solution. In this lab, it's important to know that vinegar is an acid. Test tube C is getting 1 milliliter of milk and 1 milliliter of a heated enzyme solution that I've heated before this lab. Test tube D is getting a sucrose solution and 1 milliliter of enzyme. Now I'm going to put about 5 drops of this Benedict solution into each test tube. Benedict solution changes color when glucose is present. So if there's glucose, it's going to turn from blue to yellow when I put it in heat. Now I have to heat my test tubes to check for a color change. If the color changes, then we mark the result as positive for the presence of glucose, which means the enzyme lactase is doing its job. All right, let's analyze the results. Here's test tube A. Here's test tube B. Here's C. And here's D. Remember, a color change means that the result is positive. This is what the color looked like to start with. And that's it. So I'm going to clean up things here, and you're going to work on your conclusion questions, and that's it for this lab. Great job.